In today's video, Nathan's going to show us how to extract microbial DNA from an environmental sample. This will be a sediment sample that we've collected from the Sydney Harbour estuary. DNA extraction is the first important process in any molecular biology approach, and you can do this in many different ways. Today we're going to use a commercial kit, the Power Soil DNA Extraction Kit.
First, we need to weigh a known amount from our sediment sample using a laboratory balance. Today we'll be measuring 0.3 grams for our DNA extraction.
When you're ready.
The next step involves putting your sample into a bead beading tube. This tube contains beads which will be used to mechanically break apart the cells to release the DNA.
I'm good.
The next thing we'll do is add our C1 solution. This solution contains SDS and other anionic detergents which help to break apart the fatty acids in the cell membrane. We'll pipette 60 microliters of this C1 solution into our sample. We'll then put the sample onto the vortex for 10 minutes, and this will help to break apart the cells further and to homogenize the sample as the DNA gets released from the cells. After vortexing for 10 minutes, we then put our sample in the centrifuge for 30 seconds at 10,000 Gs. So we'll next take our sample from the centrifuge, being careful to retain the separation of the supernatant and the pellet. We'll take 400 microliters of the supernatant and transfer it to a clean Eppendorf tube. Next, we need to remove non-DNA cellular material from our sample. This includes using reagents to remove organic and inorganic materials such as humic substances, cell debris, and proteins. To do this, we'll pipette 200 microliters of our C2 solution into our sample. Vortex, and then incubate on ice. After incubating the sample for 5 minutes, we'll then centrifuge for 30 seconds, 10,000 Gs. Now, avoiding the pellet, we'll transfer 600 microliters of the supernatant into a clean tube. I'll emphasize that we want to avoid the pellet because this contains all of our non-DNA cellular material. Next, we need to remove chemicals that inhibit downstream processes like PCR. To do this, we'll use our inhibitor remover reagent, which removes inorganic materials from our sample. We'll pipette 250 microliters into our sample. Vortex, and then place on ice. We'll then centrifuge the sample at 10,000 Gs for 1 minute. We'll We'll then transfer 750 microliters of the supernatant into a fresh tube. It's important again to avoid the pellet, as this pellet contains PCR inhibitors. Next, we retain our DNA by binding it to a silicon filter. To do this, we'll add 1 ml of a high salt concentration reagent and then vortex. At high salt concentrations, DNA binds tightly to silica, and this will help us to remove other non-DNA materials. Here we have a silicon filter spin column. The top part is the silicon filter and the bottom part is the collection column. We'll transfer 675 microliters of our sample onto the filter. And then centrifuge at 10,000 Gs for 1 minute. The DNA has now selectively bound to the silica membrane on the filter. We can see the collection tube contains all the other contaminants, which we'll discard. Next, we'll wash the DNA using an ethanol solution. This solution will help remove residual salts and any other potential contaminants. We'll add 500 microliters of this solution to our sample. We'll then centrifuge for 1 minute at 10,000 Gs. Now we'll discard the flow-through. This flow-through contains non-DNA contaminants removed by the ethanol. We'll then centrifuge again at 10,000 Gs for 1 minute to remove any residual ethanol. We'll now place the filter into a fresh Eppendorf tube. We want to be careful not to splash any of the residual ethanol that's in the bottom of the collection tube onto our sample. Finally, we'll need to elute the DNA from the filter into a collection tube so it can be used for downstream processes such as PCR. To do this, we'll prepare 100 microliters of 10 millimolar tris solution, tris buffer, onto the filter, and this tris buffer releases the DNA into the collection tube. We'll then add the tube to the centrifuge and centrifuge 10,000 Gs for 1 minute. Now we'll discard the spin filter and you've successfully extracted your environmental DNA, which can be used for further downstream processes.